Good afternoon and welcome to all. So today we have taken the dark field microscopy. Next we are going to take fluorescent microscope. A fluorescent microscope, it is an optical instrument or optical microscope that uses fluorescence. So do you know about fluorescence, the particles? What do you mean by fluorescence? The substance which excite. When the light falls on certain fluorescent material, they excite. So such materials are fluorescents, fluorescent material, and the property is called fluorescence. So the fluorescent microscopy, it is mainly uses fluorescence instead of or in addition of scattering reflection. The scattering reflection we have discussed in dark field microscopy, and also the attenuation or adsorption to study the property of organic or inorganic substances. So the fluorescent microscope, it refers to any microscope that uses fluorescence to generate an image. It helps in generating an image, whether it is a more simple setup like an Epi Fluorescent Microscope or a more complicated design such as confocal microscope. So the most complicated design coming under this fluorescent microscope is confocal microscope, and it may uses the optical sectioning to get better resolution of the fluorescent image. Next is about the definition. A fluorescent microscope, it is an optical instrument that uses fluorescence and phosphorescence instead of or in addition to reflection or absorption to study the property of organic or inorganic substance. This is the definition, and the fluorescence is the emission of the fluorescence, it is the emission of light by a substance that has absorbed light or other electromagnetic radiations. While what do you mean by phosphorescence? It is a specific type of photoluminescence and it is related to fluorescence. So, unlike fluorescence, a phosphorescent material, it doesn't immediately re-emit the radiation it absorbs. So, that is the difference between the phosphorescent material and fluorescence. The phosphorescent material doesn't re-emit the radiation which it is absorbed. And this fluorescent microscopy, it was devised in early part of 20th century by August Kohler, Carl Richard and Heinrich Lehmann and others. So this is the diagram, simple diagram of a fluorescent microscopy. From the sample, we can discuss about each and every terms. So you should copy the diagram. While writing the fluorescent microscopy, you should also draw the diagram. Okay. Now the principle. Most cellular components are colorless and this cannot be clearly distinguished under microscope. So the basic premise of fluorescent microscopy, it is to stain the components with dyes. So whether which object or anything, if we are observing it under fluorescent microscopy, first you should stain the components with dyes. So for the dyes, we are using fluorescent dyes. They are also known as fluorophores or fluorochromes. And these are the molecules that absorb excitation light at a given wavelength. And after a short delay, they emit light at a longer wavelength. And the delay between this absorption and emission, it is negligible, very minute, generally on an order of nanoseconds. The emission of light can then be filtered, and this light emitted, it can be filtered from the excitation filter light to reveal. So the emitted light, this can be filtered from the excitation filter. Okay. Excitation light to reveal the location of the fluorophores. And the fluorescent microscopy, it uses a much higher intensity light to illuminate the sample. So sample is illuminated with a light with higher intensity. This light excites the fluorescent species in the sample. So this higher intensity light is required to excite the fluorescent species in the sample, which then emit the light of a longer wavelength. So the image produced, it is based on the second light source or the emission wavelength. How much light is emitted, that is the image formation. So the image will be more clear. Or the emission wavelength of the fluorescent species, and it is rather than from the light originally used to illuminate and excite the sample. So the image is, the clarity of the image depends upon the emission wavelength of the fluorescent species. Next is about the working. The light of the excitation wavelength, it is focused on the specimen through the objective lens, and the fluorescence emitted by the specimen, it is focused on the detector. So emitted fluorescence which is focused on the detector by the objective. Since most of the excitation light it can be transmitted through the specimen, only the reflected excitatory light reaches the objective together with the emitted light. Now, forms. The fluorescent microscope, it refers to any microscope that uses fluorescence, and it helps to generate an image. Whether it is more simple setup like an Epi Fluorescent Microscope or a more complicated design such as confocal microscope to get a better resolution. And the most fluorescent microscope in use are Epi Fluorescents. So Epi Fluorescent microscopes are most commonly used where excitation of the fluorophore and the detection of the fluorescence are done through the same light path, that is through the objective. The most fluorescent microscope in use are, so the most commonly used are Epi Fluorescents. And these microscopes are widely used in biology and are basis of more advanced microscopic design. So the advanced microscopic design in the fluorescent microscopy are confocal microscope and total internal reflection fluorescent microscope. These are the most advanced form of fluorescent microscopes. Now coming to the parts of the fluorescent microscope. Typical component of the fluorescent microscopes are, first one, fluorescent dye. So this fluorescent dye is very important to stain the specimen. The fluorophore, it is a fluorescent chemical compound that can re-emit the light upon light excitation, and the fluorophores typically contain several combined aromatic groups or plain or cyclic molecules with several pi bonds. Many fluorescent stains have been designed for a range of biological molecules. So the different types of fluorescent stains are used. Some of these are small molecules that are intrinsically fluorescent and bind a biological molecule of interest. Some examples are given are nucleic acid stains like DAPI, Hochst phalloidin, which is used to stain actin fibers in mammalian cells. Next is important component is light source. So the four main types of light source are used, including xenon arc lamp, mercury vapor lamp, excitation filters, lasers, and high power LEDs. Lasers are most commonly used for the complex fluorescence microscopy techniques, while xenon lamps and mercury lamps and LEDs with dichroic excitation filters are commonly used for the wide field Epi Fluorescent Microscope. Next is about excitation filter, another important part. The exciter is typically a band pass filter that passes only the wavelength absorbed by the fluorophore, thus minimizing the excitation of other sources of fluorescence and blocking the excitation light in the fluorescent emission band. Next is a dichroic mirror. It is also known as dichroic filter. It is a thin film filter. It is very accurate color filter which is used to selectively pass light of a small range of colors while reflecting other colors. The emission filter. The emitter is typically band pass, the filter that passes only the wavelengths emitted by the fluorophore and blocks all undesired light outside this band, especially the excitation light, by blocking the unwanted excitation energy, including the ultraviolet and infrared rays or the sample and the system autofluorescence, optical fibers ensures the darkest background. So that's all about the different parts in the fluorescent microscope. In the next session, we can discuss about the advantages, disadvantages and limitation of fluorescent microscope. So thank you all.